Good afternoon, everyone. My name is Varun Warrior, and it's my great pleasure to welcome David Howard over here. David Howard is a Henry Wellcome Postdoctoral Research Fellow based at King's College, London. Prior to this, he worked at a division of psychiatry at the University of Edinburgh. His primary research focus is the use of genetic data to help understand the biological etiology of depression. He's a lead analyst with the Psychiatric Genomics Consortium, and he's involved in multiple projects conducting research in the field of psychiatric genetics. David, welcome. And today, David's going to give us insights into the common genetic variant architecture of depression. What do you, David?
Great. Thank you very much for the introduction, Varun. I'll just share my screen. Thank you, everybody, for attending today. I've been looking forward to doing this talk. So, depression is one of the leading causes of global disability. Current estimates are that roughly 240 million people are currently suffering with the disorder. And if we extend out to people who are likely to experience depression at some point in their lives, the number is closer to 1.2 billion. So any tiny shift we can make in improving either our understanding of depression or the treatment options available are going to just have a huge impact. Even for the steps are really small we make, the progress that we're going to see for the population, the global population, is absolutely enormous. So depression, we just talk about depression, but actually it's a really heterogeneous disorder. So in terms of even diagnosing the disorder, we rely on multiple symptoms that we pick up. So we're looking for sort of two cardinal symptoms or core symptoms, which are typically the depressed mood or loss of interest, which is also referred to as anhedonia. But then we're also looking for additional symptoms. And some of these symptoms are opposite. So we might be looking for the third one, for example, which is significant weight loss or weight gain. We've also got problems with sleep. So it's either oversleeping or undersleeping. And so what we end up with is a multitude of different ways of diagnosing depression, and someone's calculated there's over 200 different ways a person could end up with the diagnosis for depression. So this means it's a really heterogeneous condition. It also seems to be really comorbid with a range of other traits. So, for example, psychiatric traits have all been shown to be comorbid or at least related with depression. So whether that's you end up with a diagnosis of depression earlier on in life and then you later go on to develop bipolar disorder or potentially schizophrenia. It's also been shown that there's an increased prevalence of depression amongst those with Alzheimer's disease. We also find that depression is comorbid with issues related to cardiovascular system, so CVD or cardiovascular disease and stroke also have increased incidence of depression. Cancers too, so certainly breast cancer has been shown that those with breast cancer have an increased risk of depression. And then finally, there's the sort of anthropomorphic and digestive issues or traits that are shown to be associated or at least comorbid with depression as well. Now, much of the work pre the millennium was focused on sort of family-based studies and twin-based studies, and those demonstrated that we certainly have a genetic component that is associated with depression. So what I'm going to talk about today is what that genetic component can tell us about depression. I'm going to cover lots of work from thousands of researchers, scientific researchers who've contributed to the work I described, and there's millions of participants. So it's a huge thank you to all the scientists and participants of these large studies that have contributed to my talk. As Varun mentioned, I'm currently with King's and used to be with Edinburgh, so I'll give them a nod here as well. And a huge thank you also to the Wellcome and Brain and Behaviour Research Foundation, who've provided me with funding to conduct my own research. So going back to the beginning of the sort of 2000s, typically what we do are candidate gene studies. Now, these seem sensible at the time. I mean, we only had relatively small sample sizes, so studies typically only had maybe 100 or 1,000 people in them. The cost of accessing sequence data or even genotyping an individual was actually prohibitively expensive. So what we did was focused on specific variants or particular genes And these studies became known as candidate gene studies. The one that was most commonly analysed is the one sort of on the left of this plot here, the SLC6A4, and that's a serotonin inhibitor gene. And that sort of made sense to look at. I mean, it's a gene that, potentially mediates or mitigates the response to antidepressant treatment response. And we can see that there are a number of different genes were studied there, and back in 2019 actually refuted pretty much everything that had been done during candidate gene studies, and suggesting there was absolutely no evidence for these candidate genes or candidate gene interactions that were analysed. So some people were looking at whether some of these genes were influenced with smoking and depression, or parental performance and depression, but they pretty much refuted everything. I mean, one of the genes I can see along the bottom there, the D2 gene. I mean, I think potentially they would just border it all with looking at this particular variant, not just the DRD2 receptor. And actually, I think that's one of the ones that I still see popping up in both my own work and the work of others as well. So I think that's a bit of a broad statement, the fact there's no support. I think some of these, certainly the genes are potentially important, or the variants that belong within these genes are important. Okay, so what are the alternatives to conducting these sort of candidate gene studies? Well, the next step along the sort of scientific journey was genome-wide association studies, or GWAS, as they're commonly abbreviated to. So the GWAS, we have roughly 3 billion base pairs in the human genome, and roughly 1 in every 1,000 of those, it's estimated 1 in those, are sort of polymorphic, so they're not fixed in the population. There's actually variability within the human population. And so what we'll typically do is use genotyping arrays to extract out the variants that are interesting and polymorphic, and we'll study those one by one, each in turn. So we'll just look at one gene, potentially where we've got some polymorphism there. So you might inherit maybe an A or a C, sorry, we'll say a C or a T, and because we inherit obviously one from our mum and one from our dad, we end up with a pair at each position. So what we might end up with is what we see on the x-axis of this sort of small plot here. So you might inherit 2 Cs and be the homozygote C. You might inherit a C and a T, so be the heterozygote, or you might inherit 2 Ts, so you'd be the alternative homozygote. And then on the y-axis, we've plotted a phenotype. Now, in this case, we've got a sort of quantitative phenotype, so it ranges from maybe +3 to -3. So obviously for depression, we'll use slightly different scales. We're looking at cases versus controls, but again, it's a sort of similar idea. And what we're looking to do is see if the number of maybe T alleles, or whichever allele it is we're looking at, has an influence on a trait. So if there was no influence, the sort of red line there, the line of best fit or trend line, if there was no influence on the trait, it would be sort of horizontal. However, the stronger the effect, the steeper, or the steeper the line, which would be the effect size. And then you can look at also the scatter around those plots. So actually, if the pattern is very strong and there's very little scatter, it gives you greater confidence. So to calculate our sort of estimates of an effect, to see whether a variant is significant, we take that slope, divide it through by the scatter or standard error. That would give us a sort of t statistic, and we can use that t statistic to calculate a p-value. Obviously, we're looking at multiple positions right along the genome, so then we'd also need to correct for the multiple testing burden. So we use something like a Bonferroni correction. So this will bring me on to the first study that I'm going to talk about, which was conducted by the Psychiatric Genomics Consortium. This was PGC, as they're commonly abbreviated to. So this came out in 2013 in molecular psychiatry, and they were able to access data from 9 different cohorts and had roughly 19,000 people. They analysed the effect of 1.2 million genetic variants. And here is what is commonly referred to as a Manhattan plot. I use this a couple of times, so I will spend my time here to describe exactly what they show, but these are the sort of typical results you would get from a genome-wide association study. So on the x-axis, we've got each of the chromosomes in turn, organized by base pair position. Each dot on the plot represents a genetic variant. And then on the y-axis, we've got the minus log 10 p-value. So, and that's for the association with depression. So the higher up the plot each dot goes, the stronger the association it has with depression. The horizontal red line towards the upper part of the plot, that's the genome-wide significance threshold. So what we're looking for are variants that exceed that threshold. As you can see, there are no variants that surpass that. However, they do pick up on two that are approaching that level of significance. But yeah, at this point, with 19,000 people, I'm probably going to be underpowered really to detect genome-wide significant effects. I think for me, the most interesting thing to come out of this paper was actually put away on supplementary table 22. And this was very sort of early risk profile analysis, and that's now referred to as a sort of polygenic score or polygenic risk score. So what we have in the table, so in the first column, we've got the p-value threshold from the sort of Manhattan plot. And so in the second column, we can see the number of SNPs or variants that surpass that threshold. So in the first row at the threshold of 0.0001, we can see that 42 SNPs surpass that threshold. And as you move down through the rows, you can see we use an increasing number of SNPs. So what they're doing then is using those in the first row, for example, the 42 SNPs, and taking those effect sizes of those 42 SNPs and seeing how well they can predict depression in another cohort. And so that's represented in the third column. So that's the sort of p-value. So what we're looking for are significance values there. And what they've done is highlighted using the green highlight, the most significant prediction that was possible. And you'll notice it's towards the lower part of the table. This suggests that the optimal way for predicting depression is actually to incorporate a lot more SNPs rather than just a handful. So this suggests that actually depression is a really polygenic trait compared to something such as Huntington's disease or cystic fibrosis, which are much more monogenic. So the final column there is R squared, and that's the proportion of depression that's able to be predicted from the genetic component. So even with the one they highlight in green there, we're only really predicting around 0.5% of the total phenotypic variance of depression. But I think, yeah, the take home from that is depression is actually really polygenic. So it's not just reliant on a couple of genes, is actually looks to be genes spread right across the, or variants spread right across the genome that have the greatest influence on the trait. The final statement from their paper was that, you know, they think really to get a good handle on what we've got for depression, we really need well over 100,000 cases. And so that seems to be the suggestion is we just need bigger. So the next big study that came out was conducted by the Converge Consortium, came out in 2015 in Nature. And this was actually focused on a population of Han Chinese. So it was like 10,500 Han Chinese women. And it was based with data from a hospital. So it was based within a clinical setting. And they looked only at those who had very recurrent depression, so a very severe form of the disease or disorder. And their study, again, we've got the Manhattan plot here. We do actually now start to see variants that pop above this threshold of genome-wide significance. So that's that horizontal red line there. So we've got two positions on chromosome 10 that have popped above there. So following on from the Converge Consortium, 23andMe produced a paper, and that appeared in Nature Genetics in 2016. This was back to European sample and had roughly 75,000 cases and 230,000 controls. Now, obviously, 23andMe is, you know, a large study, so rather than being able to actually get a very sort of strong phenotype, they relied on the questionnaires that they send out to their participants, and that was the question basically: have you ever had a clinical diagnosis of depression? And the Manhattan plot that they produced, this is based on their 75,000 cases, we see there's actually 5 positions there that really pop out. And I've looked those positions up again since in the sort of latest studies that are ongoing, and those still really, those are the ones that are still popping out. So it looks like we're really sort of starting to make progress and get a realistic idea of what the genetic component is that underlies depression. Now, not just producing the Manhattan plot, they also looked at items known as genetic correlations. And that's looking to see whether the genetic component, say for depression, is similar to the genetic component from a range of other disorders and traits. And it can either be positive correlated, so it could go up to 1, or you might see a negative correlation going down to -1 when it's an absolute correlation. So they looked at 6 different traits and asterisks the ones that were significant. So the first 3 weren't significant. So that's LDL cholesterol, Parkinson's disease, and Alzheimer's disease. So even though we see Alzheimer's disease having sort of comorbidity with depression, it doesn't seem to have a genetic basis. Finally, the bottom 3 on the slide there, the bipolar disorder, schizophrenia, and it was actually the PGC analysis, the major depressive disorder, all showed a positive correlation. Unsurprisingly, depression was the strongest because it's a very similar trait to what they were trying to capture in their analysis. And actually the prediction or the correlations with bipolar and schizophrenia, these seem to always be around 0.25, 0.3. So this looks a pretty robust result, and we see this quite frequently. So following on from the 23 analysis, PGC came back with their paper in 2018, and this was published in Nature Genetics, led by Naomi Ray down in Queensland. And so for this analysis, they were able to access 35 cohorts now. So that's a massive increase from the 9 they had previously, and with up to 135,000 cases and nearly 350,000 controls. In the Manhattan plot, it's really starting to look a lot more like a Manhattan skyline now, with lots of peaks. And in fact, there's 44 genetic variants there that surpass the threshold for significance. And we've got 153 genes that now pop out. They went and also were able to predict the heritability of depression, and that puts it at 9%. So in twin-based studies, the estimate is around 30%. And that is likely to incorporate, from the twin-based estimates, like to incorporate a larger spread of what's available in the genome. Whereas when we look at genetic studies, we're simply looking at what's available on sort of genotyping arrays and then imputing around those. So we are likely to be missing things, but even so, heritability of depression is around 9%. Now, they were able to improve that polygenic score prediction that I mentioned earlier. So previously, we were able to capture around 0.5%. That's now gone up to around 2% polygenic prediction. They also looked at where in the sort of human body, the genetic variants that they'd identified had the greatest effect. And this is through a sort of expression analysis. And the one that really obviously stands out there are the ones towards the top of the picture. And yeah, unsurprisingly, it's things like the brain, the cortex, hippocampus. So actually that is quite reassuring to see that, because it means the genetic component we are picking up for depression certainly seems to have this impact on the brain. So we can also use their results to look at causal effects. So this is a process referred to as Mendelian randomization. And they looked at a number of different potential factors that could put somebody at greater risk of depression. So they looked at years of education, body mass index, coronary artery disease, and schizophrenia. And then they were also able to then examine whether depression itself had an impact on those or a causal relationship with those same factors. So in the results, they were able to identify that having a lower number of years of education was causal for depression, having an increased BMI was causal for depression, and having schizophrenia was also causal for depression. And in terms of bidirectional effects, we found that depression also had a causal effect on schizophrenia. So around this sort of time, the UK Biobank had released their full 500,000 information on 500,000 people. Previous to this, they'd released about 150,000, but they made their full release around this time. And so when I moved to Edinburgh, that was sort of one of the projects I first worked on. So I'm going to talk about that one. And what we decided to do was actually really focus on what is the best way to derive a phenotype for depression. I mean, UK Biobank has a huge amount of data, and it's assessed at various time points. So we've got electronic health records in there, we've got follow-up mental health questionnaires, we've got different types of questionnaire in there, and we've also got baseline information as well. So that allows you to develop a number of different ways that you can measure depression. So we came up with 3 ways, really. The first was broad depression. This was pretty much assessed across everybody in the whole cohort as the data was collected at baseline. And it was just a simple question of, have you ever seen a GP or a psychiatrist for anxiety, nerves, tension, and depression? So it's actually going to be quite broad. I mean, you notice that the proportion of cases to controls is actually much higher, really, than we see for sort of more severe depression. But still, it's likely to be capturing a reasonable component of depression. The second definition that we used was probable major depressive disorder, and that incorporated additional information that fitted around the similar criteria that I showed earlier on in the table that for the diagnostic criteria. And that brought obviously a number of cases and controls down because we didn't have information for everybody, but that should still provide us with a relatively robust measure of major depressive disorder. And finally, as I mentioned, there's electronic health records available for UK Biobank. And so we were able to look within those records for anyone who picked up or a doctor had provided a major depressive disorder diagnosis within the hospital setting. We identified roughly 8,000 cases, and typically those cases were secondary diagnoses. So a participant had visited hospital for maybe a broken leg or a stroke, and the doctor had visited them and also picked up on the fact that they had depressive symptoms and marked up as depression as the sort of secondary diagnosis. Obviously, the controls, not everybody would end up in hospital, and not everyone who ends up in hospital with depression is going to pick up the diagnosis. So I think the controls are a little looser there, but still, it should still provide a pretty robust measure. So we looked at the genetic correlations between these different definitions and actually found they were actually really relatively high. I mean, certainly comparing with other studies, anything above 0.8 in terms of depression means you're generally capturing something quite good and quite robust there. So the broad depression seemed to perform just as well, really, as the probable major depressive disorder and the clinical definition as well. So we decided we'd probably want to make use of the broad depression phenotype, and we looked at the correlations of that broad depression phenotype with both the self-reported clinical diagnosis from 23andMe data, and also the sort of major depressive disorder definition used by PGC. And we actually found, we had really strong correlations there. So this encouraged us to actually run a meta-analysis of both the UK Biobank, 23andMe, and PGC data. And that was the sort of next paper that came out within the series. That came out in 2019 in Nature Neuroscience. And in this paper, we had roughly 250,000 cases, well over 500,000 controls. We were able to identify 102 genetic variants that are associated with depression. While we were putting the paper through a review, 23andMe came back to us and said, well, look, we've continued to collect more data. We could use this new data as a replication lookup. So we asked them, yep, go ahead, do that, be great. So they looked at these 102 genetic variants that we found to be associated with depression. And in fact, they all had an effect size in the same direction as what we found in our main analysis. So that's we were pretty shocked really with that. I mean, that's like flipping a coin over 100 times and getting the same result, or the result you hope for every single time. We found 97 of the replicated variants had a p-value of less than 0.05, so they were at least nominally significant. And even applying the really robust threshold for genome-wide significance of p-value less than 5 times 10 to the 8, half the variants was still able to surpass that threshold. So that suggests we've got a really robust now version of what the genetic component of depression is. So we repeated the genetic correlation analysis, just add in new traits and see how things have changed compared to the previous analyses. And actually, we saw many of the same ones that we saw previously, but obviously we've introduced some new ones as well. So we see the same psychiatric disorders popping out again, but also included in there are things like insomnia. And that's quite interesting because obviously insomnia is one of those diagnostic criteria that's used within depression, and it also seems to now share a sort of genetic component. So if you're genetically predisposed to insomnia, you're also going to be genetically predisposed to depression as well. As you move further down through the list, you pick on things like smoking, and then that's certainly something you see in observational studies where people who are depressed are more likely to smoke. I mean, whether that's a sort of self-medicating sort of response to depression, that's still a potential avenue. And then obviously things like lung cancer, and that's likely to be related to this smoking. As you keep moving down through the list, you find things like anthropomorphic traits, so sort of weight loss, weight change. And again, that's one of those symptoms that is often picked up as part of diagnosing depression. And then finally, as you move your way down through the blues to the oranges, which are the negative correlations. I mean, these are all significant, the ones shown on this slide. You see those years of schooling. So again, it's this negative correlation suggesting a sort of less years of schooling is genetically correlated with a major depressive diagnosis disorder. From those 102 genetic variants, we actually use not just the 102 genetic variants, but you use the genome-wide sort of results. You can actually identify which genes are associated with depression. We identified roughly 270 genes that had this influence on depression, or at least an association. And we then looked at the sort of biological pathways that these genes typically influenced. And there were 15 that were identified as significant. And again, these seem to all be really related to either the brain or potentially behavioral responses. So again, that looks like we've got a really robust measure of depression there. The final thing we did in this paper was to also try and dig into whether the genes could be informative for drug treatments. And so we were able to identify the genes that we'd found. We found 37 genes that influenced or had an interaction with 222 different drugs. And these 222 different drugs fell within 54 different drug classes. And so there's a schematic of those interactions. And so we've got the genes on the left-hand side of the image, and then the drug classifications on the right-hand side. And the one that really stands out is in bright red, so running from the top left down to the bottom right. So that's actually the DRD2 that I mentioned earlier as being potentially one of those candidate genes that have held up. So it's the dopamine receptor D2, and it interacts with the NO5 and the NO6 categories. And those are the categories that contain the typical antidepressants and anxiolytics, antipsychotic treatments that are available and commonly prescribed for mental health disorders. So again, this is an interesting observation that what we're picking up in our genetic studies is actually what typically we're finding working in the clinic. But what's also interesting there is we've got a number of other different drugs that are available that could potentially target the genes that have an influence on depressive outcomes. Obviously, that needs a lot more work, but I think this sort of method of serendipitous findings, or at least slightly driven findings, could be really useful for helping people to get better treatments for depression. So we'd obviously use this quite broad definition of depression, and during the sort of review process and even discussing within the PGC, there was feelings that should we be using a really broad definition? And so it wasn't surprising that another paper came out maybe a year after ours that was saying, well, potentially using minimal phenotyping isn't the best way to target what is major depressive disorder. So this came out in 2020 in Nature Genetics, and they looked at a number of different ways of assessing whether this minimal phenotyping was effective or not. The first was looking at heritabilities. So on the y-axis, we've got that heritabilities, and they use a number of different phenotypes. And so the orange one is the broad depression phenotype. So that's have you seen a GP or a psychiatrist for help with anxiety, nerves, tension, depression. The one in dark blue is our probable major depressive disorder one. So that's a slightly more robust definition. And then the light blue, they've coded as ICD-10, and that is the hospital one. And what they suggest should be used is the one highlighted in pink. And so that's a city-based definition. So that's using even more information that's available, but it's only available on a sort of very limited subset of people. But certainly for the heritability, they found that they could actually capture a much higher heritability there. Another avenue that they investigated was the genetic correlation, saying, yeah, potentially because we're using a slightly more broader definition, maybe the correlations are quite different. I don't think I necessarily see that here. So starting on the sort of top left, we can see how the correlations compare with the first PGC analysis. And so the blue, pretty close really to what was found originally in the PGC work. And actually their pink one is slightly lower than what you'd expect there. Comparing with the 23andMe definition, so that's the self-reported clinical diagnosis, those are all much of a muchness. The bipolar disorder, which is the third panel on the top there, the one that's slightly elevated is the probable major depressive disorder, having a slightly higher correlation with bipolar disorder. The same is true when we move to the next panel, which is autism, where the probable major depressive disorder has a slightly elevated correlation compared to the others. I mean, I don't think it's necessarily significantly different, but still it's potentially of interest. Excuse me. And then as we move on the bottom panel, we have schizophrenia on the bottom left. And again, they're all much of a muchness. Same with ADHD. I mean, there's slight elevation for the light blue. So that's the hospital clinical diagnosis of depression. Neuroticism, they're all much of a muchness, really slightly lower than actually for the ICD definition. So that's the clinical definition. And again, smoking, there isn't much there. So to me, it doesn't seem to be a noticeable difference really between the genetic correlations for the broader definitions with the much more robust definitions. The final thing they looked at was the polygenic prediction. So this is the sort of risk score prediction in another cohort. On their y-axis, they've used an area under the curve, which is just another way of explaining the proportion of variance or the predictive ability of a risk score. The one that obviously pops out as having the best prediction is the help-seeking one. So that's the very broad definition, with all the others being much of a muchness. However, they suggest that potentially this is driven by sample size. Left there, so there's roughly 330,000 people who could be assessed for help-seeking, whereas actually their their pink definition only has around 70,000 people, and so potentially that's hindering their predictive ability. And so they corrected for sample size, and what that suggests is that actually the pink is the best. However, that's still below really what is, my question is, should we really be just chucking people away? Should we not just make use of the biggest data set that's available? And that's an ongoing debate that's going on. But from speaking to others, I mean, it feels like, well, we may as well go down both avenues. We should certainly conduct, very defined, strong, robust definitions of depression and see what that tells us. But also, there's no point in ignoring the large data sets out there, such as UK Biobank, Million Veterans Programme. I mean, there's going to be more and more of these large studies, and it's extremely difficult to accurately phenotype these, 500,000 upwards participants in a really robust way. So I think we need to get both avenues going. So much of the work I've been talking about so far has been focused on European populations, and that's not great, really. I mean, this is some work that came out in 2019, and in the stacked plot on the right there, we've got the global population. So we've got roughly 1 billion Europeans, 2 billion East Asians, 2 billion South Asians, and obviously you can see the rest broken down. So that's the global population. In the time series in the middle there, we've got the proportion of individuals or ancestries that are included within genetic studies. And even though the Europeans only make up 16–17% of the global population, they make up 80 to 90% of the individuals that go into these genetic studies. And what this does is it sort of biases the results and makes them only really useful for European populations. So that was taken on board by the sort of Psychiatric Genomics Consortium, or the Psychiatric Genetics community. And the last couple of studies I'm going to just touch on are more focused on those other ancestries. So a paper came out last year from Levy et al., published in Nature Neuroscience, and that included both the sort of studies that I've talked about previously, so the PGC, the 23andMe and UK Biobank data, but they've now included the Million Veterans Programme. So those are military personnel who are available, and there's roughly 1 million people within that data set, in the American data set there. So this pushes up our numbers again. So we've got now 340,000 cases and 800,000 controls, but we've also now got African-American individuals included. So running the Genome-wide association study separately by Ancestry, we have roughly 223 variants that were associated with depression in Europeans. But again, the African-American cohort looks to be underpowered, that we've seen with using sample sizes around that number, we're typically underpowered, and didn't identify any variants. But actually combining both the European and African-American samples to run the Genome-wide study, we actually find this does increase the number of variants that we can identify. So that's brought it up to roughly 230 variants. Another really interesting thing I thought they'd done in their paper looked at sort of predicted gene expression. So in each of these plots, we can see the gene expression of a particular gene. So in each box, is a gene and the gene expression, and actually where it's expressed within the brain. So we're not just focusing on the brain as a whole, we're actually starting to break it down. So if we can try and identify the sort of drugs that potentially could target specific genes that might aimed at a particular region of the brain, I think could be really useful going forwards. Okay, and so following on from this, during this last year as well, a paper came out in JAMA Psychiatry, and again, it's looking at depression, and this time in East Asian ancestries. So they had roughly 16,000 cases and 180,000 controls. And here is their Manhattan plot. And we can see there's only one variant that creeps over the line there, the horizontal red line. And compared to the earlier study by the Converge Consortium, we've actually lost the two that appeared on chromosome 10. So it's not completely clear why that's happened. I mean, there is some work ongoing with that, looking to see whether it was potentially it was the Han Chinese population that only had chromosome 10 that was significant, or potentially it was this more recurrent depression that the previous study had looked at. But one thing they certainly noticed was actually the genetic architecture identified in East Asian ancestries was actually really different to what we found in the European ancestries. So they plotted out the effect sizes between the Europeans and the East Asian ancestries. So on the x-axis are the 102 variants from my paper in 2019. So those are the effect sizes. So obviously you can see the ones that are significant are the ones that are disparate from zero or far away from zero. So they're the ones sort of left and right. And then on the y-axis, they put the effect sizes they saw in East Asian ancestry analysis. And actually there seems to be, there's a tiny sort of correlation there. There's a bit of a slope there, but it really suggests that the genetic architecture is very different for depression in East Asians compared to Europeans. Yeah, so I thought I'd pull out the same data actually and have a look how it compared within Europeans. So again, I've got the 102 variants on the x-axis. I've got the results from the 102 variants for their effect sizes. On the y-axis, I've put that replication sample that I mentioned as well from 23andMe, and you see a much stronger correlation there as a comparison between the two across co-ancestries. Another interesting thing that was included within their paper is looking at the genetic correlations that are different between the ancestries. So on the left-hand side, we can see the East Asian ancestries. On the right-hand side, we can see the genetic correlations within the European ancestries. Now, for the top four, which are generally mental health traits, those all seem fairly robust. They're all in the same direction. However, as you come down to things like body mass index, we find a negative genetic correlation in East Asian ancestries and a positive genetic correlation in European ancestries. And this is actually sort of reflected in observational studies, where typically those East Asian individuals who have lower BMIs are at greater risk of depression. Whereas in European ancestries, typically a higher BMI is associated with an increased risk of depression. Now, these are likely societal or socioeconomic factors that are interplaying here, but it does mean it's really important if we're trying to understand what's happening within a particular ancestry is to really get a sense of the societal differences between them. So certainly we couldn't, apply what we learned in a European population in an East Asian ancestry. And also then potentially if we decide to just combine these ancestries together and analyze them, what sort of correlations are we going to be losing? How are we going to be biasing our results? So I think that's sort of quite an interesting takeaway from that paper. So I'd just like to thank everyone here for listening today. I'm happy to answer any questions straight away. I mean, if you think of anything later on, please just reach out to me. My email address is down the bottom there. So thank you very much, and I look forward to any questions.
Thanks so much, David. That was a really nice talk, a very nice overview of all the amazing progress that the depression GWAS community has made.